Hello everyone. Today we are going to discuss about body fluid compartments, general physiologies. Okay. So total body water is nearly about 60% of the body weight. 60% of the body weight. And the total body water can be divided into two categories: extracellular fluid ECF and intracellular fluid ICF. So 1/3 of the total body water is called ECF and 2/3 of the total body water is constituted by the ICF. Okay. Now the ECF is again divided into interstitial fluid, plasma. Interstitial fluid and plasma comes under extracellular fluid. Okay. So 3/4 of the extracellular fluid is mainly interstitial fluid and 1/4 of extracellular fluid is mainly constituted by plasma. Now there's for pretty calculation we can say plasma is nearly 5% of body weight. And if you take about blood, it is nearly 8% of our body weight. Okay. So this is the thing which you have to keep it in mind. Then how do we measure the fluids in these compartments? Okay. So the measurements. So basically it's measured by the method which is called as dye dilution method by Stuart Hamilton. So basically it is calculated by Stuart Hamilton dye dilution method. Okay. So now what is the method? Basically there are two requirements which should be fulfilled in this particular method. One, the dye should be evenly distributed in the compartment. The dye which we introduce, it should be evenly distributed in the compartment. Secondly, the compartments, the dye should not escape or leave from the compartment. The dye should not leave from the compartment. And secondly, the dye should evenly get distributed in the compartment. If these two parameters are being fulfilled, then only we use this particular method. Okay. So basically the volume of ECF is calculated by the initial volume of dye injected in the compartment by the concentration of the dye. So this is the particular formula. V equal to volume of ECF, extracellular fluid. I is the dye injected and C is the concentration of the dye. Now in a case if the dye leaves the compartment, then we can modify the formula into V equal to I minus A by C, where A is the amount of dye left the compartment. Now let's discuss about the dyes which we use for measurements. Okay, so let's talk about total body water, which is the dye used. Basically, the dye used here is deuterium oxide. Deuterium oxide, D2O, and then tritium is also used. Tritium is also used. Antipyrine is also used. Is also used. And as we have deuterium is used to calculate the body fluids of total body water, the total body water is also called as deuterium space. Now let's talk about extracellular fluid. The dye used here are basically saccharides which cannot be metabolized, non-metabolized saccharides, and basically sucrose is used, then inulin is used, mannitol is used. The thing which we have to keep it in mind is that the best dye to measure the ECF, it is inulin. Inulin is the best dye used to measure ECF. Why? Because it does not get metabolized easily and does not leave the compartment, whereas we have called deuterium space for total body weight. Here the extracellular fluid ECF is called as sucrose space. Now we have to calculate the intracellular fluid. So to calculate the intracellular fluid, it is simple. We just have to take the total body water and minus ECF from it, and we will get the ICF. And in case of calculating the plasma, here we use radio labeled albumin, which is radio labeled iodized albumin, or you can just say radio labeled iodine is used. Then in case of red cells, RBC, chromium tagging is being used. So these are the things here we have discussed. The body fluid compartments, measurements, and the dye used. Now the next topic which we will be discussing is the disturbance faced in the body fluid compartments. Okay.